Hello, welcome to Introductory Molecular Biology and Diagnostics. This is continuation of Lecture 5 and 6. Now to our objectives. Like you are aware, we first dealt with molecular biology and now we are dealing with molecular diagnostics with respect to the history of molecular diagnostics and introduction to all these that has been done in the previous lesson. In this lesson, we'll be looking specifically at nucleic acid techniques. We'll be talking about DNA restriction, how DNA is cut. We're looking at the nucleic acid analysis, how we can analyze them. We're looking at amplification of DNA, how we can multiply a particular gene of interest. We're looking at sequencing, how to know the particular nucleotide arrangement in a gene. Then we'll come to hybridization and expression analysis. Then we'll briefly talk about molecular cloning, how we do recombinant DNA technology. And then we'll be quantifying DNA and RNA. Now let's talk about molecular diagnostics. So as for the history, it's been given to you as an assignment and I'll be giving you your scheduled submission time. So molecular diagnostic testing combines laboratory testing with the precision that molecular biology brings into making laboratory diagnosis in our day-to-day activities as laboratorians, viral testing, microbial, genome testing, and etc. very successful and possible. Now, like I keep saying, molecular diagnostics laboratory testing is becoming useful in so many jurisdictions. Here, we might be a little slow in implementing because of cost, but in many other jurisdictions, our day-to-day infectious disease testing, clinical chemistry testing, monitoring of disease and everything is done by molecular diagnostics because it is more specific and the precision and the speed of result output is very, very, very high. So advancement in molecular diagnostic testing will continue to improve the accuracy and speed by which we can detect microbial pathogens or analyze the patient's genes. And it's becoming an essential aspect of patient-tailored interventions and therapeutics. So we are seeing a future where when we are doing disease treatment, it will be very specific. So patient A has diabetes, patient B has diabetes, but by the basics that we have from, or the knowledge we have from molecular diagnostics, we're able to treat patient A differently and patient B differently, just like we do in our regular microbiology labs. We do sense. So for a particular antibiotic or for a particular microbe, we use two different antibiotics depending on our zones of inhibition we measure for each of the samples we received. So the same thing is what molecular diagnostics is bringing in general in all aspects of patient treatment. Now, in general, molecular diagnostics doesn't only depend on the nucleic acid techniques, which we are going to talk about, but there are also protein techniques and then cell culture techniques. So, with protein techniques, we are using our proteins to determine our disease, and in cell culture techniques, we culture the cells to determine our disease. Now, there are various sites and samples that can be used for molecular diagnostic studies. So in nucleic acid techniques for molecular diagnostics, we look at DNA amplification and sequencing, such as the PCR tests procedures. Then we look at RT-PCR, real-time PCR and its applications, various techniques for blotting, molecular cloning, and then DNA quantification. Now in protein techniques, we'll be looking at various techniques that are relevant in uh, identifying the functions and the structure of proteins. We are able to identify the function of proteins when we're able to determine its structure. We're able to identify the function of proteins when we're able to express a particular gene's output with respect to its proteins. Now, this has numerous applications like producing hormones like insulin and the rest. So that is what protein techniques with respect to molecular diagnostics looks at. Now, we are clear in mind that the solubility, functionality, speed, and yield of these proteins are very relevant in choosing a mode of expressing these proteins. Then we'll talk about cell culture techniques. There are numerous or various types of cells that are grown in our labs. If you take a microbiology lab, you look at various microbes that come to your lab for testing. When we come to oncology, where there's an abnormal growth of cells, that is also looked at. Now, the term tissue culture is often a generic term which refers both to organ culture and cell culture. So you can be culturing something from the organ or culturing cells or individual cells. Now, cell cultures are derived from either primary tissues or cell suspensions. So you pick directly from the tissue, or there's a solution where the cells are found in there. Now, the primary cell cultures have a finite lifespan, so they live for a period and then they die off. That is in culture. And then we have also continuous cell lines, which are usually abnormal cells and they do not seem to have a lifespan. They keep moving from one form to another. A typical example is regular cancerous cells. Now, cell culture is a technique that involves isolation. So you take it out and maintenance in vitro of cells isolated from tissues or whole organs that are derived from animals, microbes, or plants. So by definition, you first need to isolate your cells and then maintain it in vitro. And like I mentioned the previous time, you need the right conditions to sustain an in vitro uh, tests of cells. Now, animal cells have more complex nutritional requirements and keeping them is difficult. So you need stringent conditions for their growth and maintenance. By comparison, microbes and plants require less rigorous conditions and grow effectively with a minimum of needs. Regardless, practical cell culture is governed by the same general principles, requiring a sterile, pure culture of cells, use of appropriate aseptic techniques, and suitable conditions for optimal growth. So you have uh, key needs which every in vitro cell culture should have. So pure culture of cells. So what you are picking shouldn't be made up of different kinds of cells to confuse your growth, like missed growth that you are having. You also need to observe aseptic techniques, ensuring that no contamination takes place. And as usual, you need your suitable optimal conditions to make sure that your cells are growing successfully. Now, once you establish your cells in culture, so many things can be done with it. For instance, it's a way of studying intracellular processes, including protein synthesis. So you want to find out how proteins are produced with or from particular cells. You pick them out, give them the right conditions, and then they will start behaving the same way like they do for you. It can help in signal transduction mechanisms and then also drug metabolism. Now, it's one of the ways that various actions like interactions in genetic cell-cell interactions take place, and then interactions producing various genetic outputs are also done. You can also study drug impact on various cell lines. So for instance, we produce a drug that has an impact on, say, the pancreas. It is treating an ailment, but I want to see its impact on the pancreas. You can pick cells from the pancreas, give it an in vitro environment, then present it to the drugs and see how it would affect it. Or we have a drug that is able, someone claims to have an ability to treat, say, cancer. You present the cancer cells with the drug and see its impact. Now, in medicine, it can also be used to study genetic abnormalities using various chromosomal analysis of these cells. So if a mother is going to give birth and we are afraid that there might be some genetic abnormalities, you can even study it ahead of time. Now, in the same way, we can also use it in studying viral infections, both in terms of qualitative studies and quantitative studies of viruses. So we come back to our nucleic acid techniques. So let's first talk about DNA restriction. Now, the process of DNA restriction involves the cutting of the DNA at specific sites. So you are cutting, but not just cutting randomly, you are cutting at particular points. Now, the enzyme restriction endonuclease that we obtain from bacteria is what we use as a DNA restriction enzyme or restriction endonuclease. Now, these cuts DNA at specific sites. So there are particular points along the DNA molecule where the cutting is obtained. Now, the next point talks about the source of this restriction endonucleases or these DNA restriction enzymes. Now, in bacteria, these restriction enzymes cut through foreign DNA. So if there's an invading foreign DNA, bacterial cells have restriction enzymes which cut through. Now, these restriction enzymes is what scientists have been able to, shall I say, scoop out and then manipulated it for use in DNA restriction. So these genes are known to have the ability to cut DNA or foreign DNA at particular points. Now, these restriction enzymes commonly used in the lab have 4 to 6 base pairs or recognition sites or binding or cutting sites on the DNA. So it's able to identify these cutting sites on the DNA. Now, any 4 to 6 base pair nucleotide sequence occurs at random in a sufficiently long stretch of DNA. So if you take a DNA, it will be cutting between 4 to 6 nucleotides or base pairs So as you go along the DNA, when it sees a restriction site within this number of base pairs, then it cuts it. So restriction sites occur naturally in the DNA. That is very key to know. It's not the restriction enzyme that is causing the restriction sites to occur. In every DNA, there's a restriction site. Restriction site mapping is the determining of where in the DNA sequence a particular restriction enzyme recognition is located. So if you want to do restriction site mapping, then you are determining which particular points on the DNA has the restriction site located. Then you can do a DNA restriction site mapping the points where cutting can take place. So restriction enzymes are the backbone reagents of cloning. When it comes to cloning, without restriction enzymes, you cannot continue, because you need to have particular points you are going to cut. And when you cut those points, you should be able to join. So these restriction enzymes are backbone reagents of cloning, but there are other applications of it: DNA fingerprinting, genetic identity epidemiology, and blotting techniques. They all require the restriction enzymes. Although we think of them more in cloning, all these other areas require these restriction enzymes, cutting at restriction sites. Now, in nucleic acid studies, one of the major hurdles then was how to obtain a large number of a particular genetic code or nucleotide sequence to be able to study it. So let's say I am able to identify a particular gene that is coding for, say, good eyesight. Now, if I want to study that, how well can I get enough of such a sample to study? And that is where DNA amplification comes in. Now, in former times or in early analysis of the nucleic acid, scientists were having a limitation of the availability of the material to be analyzed. How can you generate enough copies of a single gene for you to study? Now, and if you wanted to do that through cells in culture, then you have big work to do, because then you'd have to find a way of getting more of such to study. So your options are either one, having a large number of the raw sample and isolating your nucleic acids or DNA from it, or the ability to multiply that particular gene using cloning, and then with the bacterial plasmids you're able to harvest many of such, and then you take out your DNA. So the complexity is one, getting more of the sample, or two, multiplying an identified gene within a bacterial plasmid. That is through cloning. So if you want a large sample, or then if you wanted a large sample of a particular gene, you either use these two methods, either by using large sample and extracting your DNA, or when you cut out the particular DNA of interest, you push it into, or you clone it into a bacteria, cause the bacteria to multiply severally, then you do your isolation from the bacterial cells again, and that was laborious. Now, the advent of the ability to amplify a specific DNA sequence opened the ability to analyze at the nucleotide level virtually any piece of DNA in nature. So at the point scientists succeeded in amplifying a particular DNA, and that was when studying the nucleotide became very easy. Now, the first specific amplification method that was identified as the PCR, the polymerase chain reaction. Polymerase chain reaction. Other kinds of amplification methods exist, but they are all dependent on the original concept of the polymerase chain reaction. Now, there are different methods that have been developed to amplify nucleic acids, and we can break these down into target amplification, the probe amplification, or the signal amplification. So these are the three key kinds of nucleic acid amplification that exists. Now, with target amplification, we have a particular target sequence that we would want to amplify or want to multiply. So the amplification of nucleic acids by target amplification involves making copies of a target sequence. So you have a particular target that you want to multiply to such a level as being in millions of copies and making it possible to detect them in vitro. Now, this is analogous to growing cells in culture and allowing the cells to replicate their nucleic acid as well as themselves, so that you can visualize it on an agar. And like I said earlier, if you need a particular target, lots of that, you'd have to either multiply that cells, that portion of the sequence in a bacterial plasmid, and then harvest it, or get a larger sample and extract your DNA from that. But in target amplification, we know a particular portion of the DNA that we need. We multiply that target severally. The next point is that the difference is that waiting for cells to replicate to detectable levels can take days to weeks or months, whereas replicating the nucleic acid in vitro only takes hours. So the advantage here is time. If you want to study a particular portion, you can't be waiting for months or weeks or days before you can get your target sequence. But with PCR, you're able to identify that portion, multiply that portion in hours, and then take it out for use. So that's what the last point is saying. So PCR is the first and prototypical method for amplifying target nucleic acid. Now there are other ways of going about transcription-based amplification, etc., etc. PCR is the first that was found. So let's talk about PCR. So you've heard PCR, PCR, PCR. So what's PCR? So PCR is a method of generating many copies of a specific DNA sequence. Now, newer technologies and sample preparation applications permit the use of PCR in a more timely manner than some conventional technologies. So you're able to use PCR in a more timely manner than other conventional technologies. Now, the amount of target nucleic acid in the sample may be extremely small. We're still talking about the use of the PCR, such as in forensics and prenatal testing, or when the quality of the DNA sample is poor. Yet, PCR may yield results not obtainable from other techniques. You go to a crime scene, you see a blood spot, you want to find out whose blood that is. That might be just a drop of blood. And out of that drop of blood, you will get less DNA for your analysis. Now, with PCR, that small blood, that small hair, or that small fluid you pick from a pregnant woman can be multiplied severally, and then studying it makes it very simple. Now, this PCR procedure will amplify picograms of nucleic acids to micrograms in a short period of time. So you have a picogram, in a short time you have micrograms of that same nucleic acid. Now, because of the specificity and rapidity of the testing, PCR has just about replaced every other nucleic acid procedure in clinical labs. So there are so many other procedures, but the use of the PCR has become a major stake in our laboratory testing. Now, let's talk about requirements for the PCR. The first requirement for PCR is the DNA polymerase. Remember DNA polymerase? When we are talking about the DNA earlier on, how the DNA is able to replicate itself, you need a DNA polymerase. Now, the DNA polymerase or DNA pole is the enzyme that incorporates nucleotides onto the end of the replicating DNA strand. So that's DNA polymerase for you. Now, this DNA polymerase that is used in our PCR is a specific one that has been isolated from the organism Thermus aquaticus. So you hear us talking about TAC, T-A-Q, Thermus aquaticus, T from Thermus, and then A-Q from aquaticus, Thermus aquaticus. Now this is a DNA polymerase that has been taken from Thermus aquaticus because it is heat stable, able to withstand heat to about 95 degrees and then cooling and still function. You remember that these are proteins that have issues when it comes to heating, but Thermus aquaticus polymerase has been identified to be heat stable, able to withstand heat to 95 degrees and then dropping during your PCR procedures. So that is one of the requirements. You need a DNA polymerase. Now, the DNA polymerase needs a DNA template. It also needs all 4 nucleotides because you are going to form, or let me say, you are going to copy or replicate a particular DNA strand or section. So you need your nucleotide because it's the nucleotide that forms your DNA. So all the 4 nucleotides will be relevant here. Then you will need buffers. Then the buffer is going to help you have the relevant environment for your reactions. Then you need primers. Primers, that's free 3' hydroxide groups or primers. And you know, primers are what begins the whole process of the DNA replication. Now it's onto these primers that the nucleotides are added. So it gives the starting point for the reaction to continue. Now, the components of the buffer can either be monovalent or divalent cations, which will provide the optimal conditions for accurate and efficient replication. That's the use of the buffer in there. So your requirements again, you need a DNA polymerase and then a template. Now, the template is your original strand that you are going to replicate. You need all 4 nucleotides because you are going to replicate not just anything but a nucleotide arrangement. So all those 4 sources should be available. When you need guanine, it should be available. When you need T, it should be available. When you need A, it should be available. When you need C, it should be available. So all these 4 should be available. You need buffers to give the requisite environment that the replication or the polymerase reaction can take place, and then your primers, which would actually start the whole copying of the required DNA. Now let's look at the primers. Now the next useful thing we are looking at is the primers, like we mentioned in the other slide. Now the primers are critical because they determine the specificity of the PCR. Without a primer, you can't copy anything. It is what is required for you to know which portion of the DNA is supposed to be copied. Now, the primers are said to be analogous to probes. We'll come to the probes later in blotting and hybridization procedures. Now, these primers are chemically manufactured in a DNA synthesizer. They're able to manufacture, or they are manufactured for studies. Now, for a DNA, you have a strand, a double helix. When this is open, you have two strands. Now, if you want to copy that portion, you will need that. If you place it down, that there's a top portion, there's a down portion. You need to copy the top, you need to copy the down. So this is what your primers do. The forward primer must bind to the target DNA sequence in the 5' direction. Then the reverse primer, which is on the opposite strand, would also bind at the 5'. So there's a particular portion you want to copy. On the left hand side, if the 5' is there, the forward primer binds there. Then on the lower strand, the other 5' end has to also be bound. So by the time they all move, the portion of the DNA that you want to replicate has been produced. So in designing the primer, there's the need to have a knowledge of the target sequence. You can't just produce the primer without knowing the target sequence. So there's the need to know how that target sequence is. So for instance, if you want to find out about a particular, say, bacteria or parasite, there's a particular portion of the parasite that is known, and that is where the primers are going to bind to. So in producing your primer, you know that the primer is going to bind to, say, 0.114 kilobyte on the DNA strand of the parasite. So there's the need to have an idea of this particular DNA you want to run a PCR on. Now, DNA sequencing also has, or DNA sequencing in general is identifying the nucleotide arrangement. And now in DNA sequencing, the same thing is done like in PCR, but it uses one primer and replicates one strand, whereas PCR uses two primers, one moving in this direction, one moving in the opposite direction, and then flanking the target sequence, replicating both strands at the same time. In sequencing, one primer, one strand replicated. In PCR, two primers, and then the two strands being replicated. That's the difference between PCR and then DNA. Now, let's look at the steps in PCR. In doing your PCR, you first denature, then you anneal, then you extend. So denature, anneal, and extend. Now the first step of denaturing. So in denaturing, the target DNA is denatured. You know denaturing. It is melted by heating it up to more equal to 95 degrees Celsius. And at this temperature, the strands stay melted, or they stay opened up until you lower this temperature. And at this point is where the base pairing can take place, because you need to open it up before you can do that. So you unzip like we are going to do DNA replication. Now the second step is annealing. Now during this period, a pair of primers is added in excess quantity to outcompete the complementary strand for binding to their target region. So when you open up the strand, you bring in more primers, and these primers bind to the required position. Once it is bound to it strongly, the complementary strand cannot come back to close up the unzipped DNA portion. It comes to outcompete the complementary strand for binding to their target region. Now, these primers are synthetic oligonucleotides which are constructed to bind or anneal to known sequences. And like we mentioned earlier, it flanks either side of the target region. So one binds to the top, one binds to the down. Now the temperature that is needed for this binding or annealing is between 40 to 70 degrees. Between 40 to 70 degrees. Then the third step is the extension. Once the primers have bound to their requisite position, then your DNA polymerase is added to the reaction. And this the DNA polymerase is going to identify your primer and then copy the strand of interest. So the DNA polymerase mimics the natural replication of the DNA and copies an adjacent DNA template to extend across between the primers. So a primer is bound to the one end, bound to the other one. So the polymerase moves along the stretch and replicates that portion of the DNA, like in regular DNA multiplication or replication. Now the extension temperature is commonly 72 degrees, and it is based on the type of DNA polymerase used. That's the temperature that is being used is based on the type of DNA polymerase that is being used. Now we come to the cycle. When we talk about the cycle, we are talking about the heating conditions that are available for this whole process to take place. Now, like I said, the major step during the whole process of the PCR is the heating cycle. We're talking about some 95 degrees Celsius, talking about 40-70, talking about 72. In the PCR procedure, there's a change in heat. This is relevant, like I said earlier, in the PCR steps in denaturing, annealing, and then extending. So heat is applied to denatured DNA again, followed by cooling to allow more primer to anneal. So when heat comes, it denatures. When it is cooled, the primer is able to anneal or bind, and then the extension takes place by the polymerase. Now this reaction takes place in a program system which we call the thermocycler. Now this is programmed to give rapid and accurate temperature changes because the PCR procedure is a chain reaction. That is the name, polymerase chain reaction. So the polymerase action, the DNA polymerase occurs in a chain. So it produces to the end, and it starts the process again, denature, anneal, extend. Now in this thermocycling procedure, the number of cycles may vary from one to the other, but usually it's between 30 to 35 cycles that are performed. So the cooling, the heating repeats itself between 30 to 35 times. And then for every one template that you have within there, 235 copies are produced. So if you have 100 copies, we are producing 235 from each, then you can imagine that 100 and each is 235, you will have 235 of each produced. That's like 23,000. 100 more, 235,000 copies more of each has been produced. Now within 1 to 2 hours, PCR can produce millions of copies, which we are calling amplicons of the DNA. But if you want to do the same in vivo, it will take a very long time. Now the real advantage of the PCR is the ability to amplify specific targets. So we pick a particular section and we reproduce it. Just as the southern blot first allowed analysis of specific regions in a complex background. We'll come to the southern blot later. PCR presents the opportunity to amplify and essentially clone the target sequences, the specified areas. The amplified target then can be subjected to innumerable analytical procedures, and I think this has been overstated. A particular portion you want to replicate. When you replicate it more, then you're able to utilize it for your desired action. So lots of DNA amplicons produced makes your work feasible. Now there are modifications to the PCR. So the PCR has undergone different modifications to satisfy particular, should I say, requests and desires. Now the available modifications are the multiplex PCR, that's for detecting multiple targets in a single run. So for a particular DNA, you are not looking at just a single target. There are a number of targets you are looking at. So multiplex PCR. Then you have the reverse transcriptase. The reverse transcriptase, that is using the PCR on RNA template instead of DNA template. So reverse transcriptase. You remember transcription. Transcription is an event using the RNA. So reverse transcriptase PCR is also another modification. Then we have the nested PCR. The nested PCR. Now these are more like amplified products being used as templates. So you have amplified a particular product, and now they have become the template on which you are relying for your studies. And then another modification is quantitating of the PCR. That is quantitative PCR or real-time PCR. Any of these can or are modifications of the regular PCR that are done. So we'll look at these briefly, how each of these is. Now the first modification we look at is the multiplex PCR. More than one primer pair is added to a PCR so that multiple amplifications are primed simultaneously. You are doing the PCR, but you're not concerned about just one particular chain reaction, but then you have a complex, so you have more than one primer in there. So resulting in the formation of multiple products. So your output will not contain only one particular portion of the gene replicated, but series of them. Multiplex PCR is especially useful in typing or identification analysis. So if you are typing, you want to compare an organism to another, you can't just use one portion to compare. So you need to look at different sections, multiply different sections, and then you can compare. Individual organisms from viruses to humans can be identified or typed by observing a set of several PCR products at once. So that's what I said earlier. Now, multiple organisms have been the target of multiplex PCR in clinical microbiology laboratories. So that one respiratory sample, for example, can be used to test for the presence of more than one respiratory virus. So someone has a respiratory infection, will not just zoom in looking at just a single organism, but we'll look at all the possible respiratory organisms that can cause that infection. So at the end of the PCR, we would have found out the particular one that is available, not just concern ourselves with one particular virus and then ending up before we think of testing for the other. Concurrently, at the same time, we're able to run the test for all these organisms. Then we come to the reverse transcriptase. And like I mentioned, transcription is about the RNA. So nucleic acid amplification by PCR requires a double-stranded DNA template. That is a regular thing. If the starting material for a procedure is RNA, it must first be converted to double-stranded DNA. Procedure requires double-stranded DNA, and you are starting with an RNA. So your first step is to convert the RNA to DNA, as a double-stranded DNA. And this is accomplished through the action of the reverse transcriptase, an enzyme isolated from RNA viruses. So in RNA viruses, they have the reverse transcriptase, which moves RNA to DNA. You know the transcription process, or the transcriptase, that's DNA to RNA, but the reverse transcriptase, that's RNA to DNA. This enzyme first copies the RNA single strand into an RNA DNA hybrid. So a double strand is an RNA DNA hybrid strand. And then uses a hairpin formation on the end of the newly synthesized DNA strand to prime synthesis of the homologous DNA strand, replacing the original RNA in the hybrid. So the RNA portion is replaced to another DNA strand. The resulting double-stranded DNA is called cDNA, that is copy DNA or complementary DNA. So the new DNA strand, which originally was an RNA, has been replaced, is the copy DNA strand. Now, this product is adequate for PCR to take place. So you have formed a double-stranded DNA, that is the complementary DNA, which has been done from an RNA. Now, this product is adequate for your PCR. Reverse transcripted PCR is used to measure RNA expression profiles, to detect rRNA, to analyze gene regions interrupted by long introns, and to detect microorganisms with RNA genomes. So if you have a microorganism that has RNA, this is how you are able to identify it. You can also detect the presence of rRNA in samples. So this reverse transcriptase, moving from DNA, RNA to DNA, moving from RNA to DNA. Then we have the nested PCR, the nested PCR. So in this, it's a modification that increases the sensitivity, one, and then a specificity of PCR test. Because regular PCR presents a low level of target and can have interfering sequences which can affect the reliability required for clinical application. So in regular PCR, you produce a longer target region, but in the nested, you zoom into the particular portion of what you have produced originally in a regular PCR. So you produce a regular PCR, you pick another portion of that thing you've produced, the amplicon, and then work on just that section. So in the nested PCR, two pairs of primers are used to amplify a single target into two separate PCR runs. So the first PCR goes, then the second pair of primers designed to bind slightly inside of the binding sites of the first pair. So like I said, you produce a longer replicated portion, then you do a second PCR. Now this second PCR is now going to work on the first thing you have replicated. So you had a DNA, you have produced a portion of the DNA, which is your target. Now you zoom into that a portion of that target and then you replicate just that small portion. So this amplifies the product of the first PCR in the second round of amplification. The second amplification will specifically increase the amount of the intended product. So the intended portion is now further replicated, and this is nested PCR. Then we come to quantitative PCR, and like the name says, quantity. So standard PCR procedures will indicate if a particular target sequence is present in a clinical sample or not. In an instance when the quantity of the target present is relevant for clinical or other use, qPCR is needed. So if you need to know the quantity, then you need to do quantitative PCR, qPCR, or real time. The advantage of this method over standard PCR is the ability to determine the amount of starting templates accurately, unlike other earlier methods. It is useful in clinical applications such as copy numbers of disease human genes, viral load, tumor load, and the effects of treatment. So someone has hepatitis B infection, you want to know the viral load. You pick a sample from the individual, but because of the quantity of the required DNA within it that you can get, you need to amplify it using PCR. When you finish your PCR, the quantity of that gene that you've replicated is not what was originally in the sample. So in quantitative PCR, it helps you know the original quantities that you had before you started your whole PCR multiplication. Then we can be talking about arbitrarily prime PCR, arbitrarily prime PCR. Now this is also known as randomly amplified polymorphic DNA or random amplification of polymorphic DNA, RAPD. Now short primers with random sequences are used to amplify arbitrary regions in the genomic DNA under low stringency conditions. So you pick a particular DNA, you try to replicate arbitrarily or generally or randomly portions of that DNA. Now with this method, PCR products are generated without knowing the sequence of the target or targeting a specific gene. So you don't target a specific gene, you just zoom in and replicate portions. It is possible with this method to obtain multiple products depending on how many times a short sequence appears in the genome. So if a short sequence is found severally in the genome, then it would also be produced severally. In traditional PCR, only one product is generally obtained. Arbitrarily primed PCR has been useful in epidemiological typing of microorganisms. So you want to see microorganisms, whether they are the same. You randomly replicate a particular portion of their genome or their DNA, do it for all the organisms, and then compare if the portions that you replicated are the same. Then you can use that one to type them. Then transcription-based amplification systems. In this, RNA is the usual target instead of DNA. A DNA copy is synthesized from the target RNA, and then transcription of the produced DNA produces million copies of RNA. So in this one, you are amplifying the RNA portion of it, or an RNA is what is the final product. So you produce millions of the RNA. Okay, so the earlier ones is target amplification. Now we move on to probe amplifications. Now in probe amplification procedures, the number of the target nucleic acid sequences in a sample is not changed as it is seen in the target amplification procedures like PCR. In PCR, it is the portion that you desire that you multiply, you amplify. But in probe amplification, it is rather the synthetic probes that are, let me say, replicated. So synthetic probes that are specific to target sequences bind to the targets where the probes themselves are amplified. So the probes that you amplify. There are three major procedures that are commercially available for probe amplification. We have the ligase chain reaction, LCR. We have the strand displacement amplification, SDA. This is not your regular SDA. And then you have your Q beta replicase. Q beta replicase. So these are the three kinds of probe amplifications we want to look at. Let's look at the LCR, the ligase chain reaction. Now, this is a method for amplifying synthetic primers or probes complementary to the target nucleic acid. LCR is better classified as a method of probe amplification rather than target amplification, as the copy number of target molecules does not change. So the target molecules are intact. It is the probe that is being amplified or replicated. So LCR is similar to PCR, but there are some few differences. Let's look at the differences. The first difference here is the entire target sequence must be known in order to prepare the oligonucleotide primers for LCR. So the final target must be known before you can produce your primers for it. The second point, there can be, in regular PCR, there can be a distance between the primers of up to hundreds of bases, which is all part of the amplified sequence. But in the LCR, the primers bind adjacent to each other. separated by only one base. So the distance is just a base difference, but in PCR you have hundreds of bases difference between the primers. You know, the primers bind on at this end and on that end, so and replicate the two strands. We are talking about the distance between them. In the LCR, we are just one base interval, but in PCR you have hundreds of base intervals. And the third difference is instead of using DNA polymerase in synthesizing the complementary DNA by the primers as in PCR, we rather use DNA ligase in the LCR. That's a ligase chain reaction. We use a DNA ligase, and it is used to ligate to bind the adjacent primers together. So this is generally about the ligase chain reaction. Now let's look at the similarity. So LCR, like PCR, requires a thermal cycler for the temperature changes that you need to drive the reaction. When temperature is cooled, the primers anneal, bind, or connect. If the complementary sequence is present, an absence prevents annealing. So if you have a complementary sequence that will bind to the two strands, it prevents the annealing, and it is key to identifying mutations. If there are mutations in structure, then this is a key step to identify that. So LCR can be used to detect point mutations in a target sequence. The point mutation that occurs in the beta globulin of patients with sickle cell disease, as compared with normal beta globulin, was first identified or was the first application of the LCR. So first used to determine the changes in the beta globulin of sickle cell patients. So generally, ligase chain reaction LCR and then polymerase chain reaction are similar by using the thermocycler, which helps raise temperature and then cool it at specified periods for the chain reactions to take place. Then we come to the SDA. Now it differs from other amplifications by being isothermal, depending more on room temperature. So you have a cycler in the other situation, this one you are isothermal. So depending on more on room temperature after the initial denaturation steps. For denaturation, you need the high temperature, but afterwards, then your temperature returns to room temperature. Like in LCR, the major amplification products are the probes, because you know we are at this point we are dealing with probe amplification. So what we are amplifying are the probes. Earlier we were talking about targets. Now we are talking about probes. So the major products are the probes or the primers, and not the product of the DNA target. So this method was first widely used in the detection of Mycobacterium tuberculosis by incorporating fluorescence polarization to detect the amplified target. We'll come to fluorescence later, but generally know that it was the method used to identify or to detect M. tuberculosis. So that's about the SDA. The SDA typically runs isothermal rather than the thermocycler running throughout the whole procedure. When we come to the Q beta replicase, now you know when you hear is the thought should come to mind what that is an enzyme. Now this is another method for amplifying probes. So you know we are talking about probe amplification that have specificity for a target sequence. The method is named for the major enzyme that is used to amplify the probe sequences. That is Q beta replicase. And this is an RNA dependent polymerase from the bacteriophage Q beta. The target nucleic acid in this assay can either be DNA, which must first be denatured, or RNA. Now, it has been used primarily to amplify the nucleic acid associated with infections or infectious organisms such as mycobacterium, chlamydia, and then HIV. So QB replicase is specific determination or used to determine the specificity for target sequence. Now, our final kind of amplifications is the signal amplifications. So signal amplification procedures differ from target amplification procedures in that the number of target sequences does not change. It looks same about probe. Instead, large amounts of signal are bound to the target sequences that are present in the sample. Because the number of target sequences does not change, signal amplification procedures are inherently better at quantitating the amount of target sequences present in the clinical sample. So in the target, the target sequencing, you are multiplying the target. But in the signal amplification, you are now binding different signals to the same target. So it's possible to measure the quantity of your target in a sample because it does not change. The quantity does not change. Now, the various kinds that are available are branch DNA amplification, hybrid capture assays, cleavage-based amplification, and then the cycling probe. So we'll deal with these. Now, in the branch DNA amplification, a series of short oligomer probes are used to capture the target nucleic acid. We first capture the target nucleic acid. Just look at the drawing. Look at this portion. See capture probes. Good. So these are short oligomer probes and are used to capture the target nucleic acid. Then additional extender probes. These are the extender probes. Bind to the target nucleic acid. So capture probes bind, extender probes also come to bind. And then to multiple reporter molecules. So multiple reporter molecules also come in. So these are such. Multiple reporter molecules also bind, loading the target nucleic acid with signal. So now the target nucleic acid is then loaded with signal. So you can now spot it when it is there. This is a branch DNA amplification. Then we have the hybrid capture. Hybrid capture assays. Now we are saying that target DNA is released from cells and binds to single-stranded RNA probes. This is recognized by antibodies which bind to them and forms a bond with an alkaline base, resulting in a chemiluminescence reaction and measurement. This is available for detection of hepatitis B and human papilloma virus. Now in the hybrid capture, the target DNA of concern is released from the cell. So you find a way of releasing that one. Then it binds to single-stranded RNA probes. Now these RNA probes are recognized by antibodies which binds to them. So antibodies bind to this. And it forms a bond with an alkaline base, resulting in a chemiluminescence reaction. So once this bondage comes in or this binding comes in, then the alkaline base is formed. then the chemiluminescence is released. Now, chemiluminescence is just creating a chemical bond that can give out a signal. So that's how the hybrid capture behaves. Target DNA from cells bound to single-stranded RNA, then recognized by antibodies. And when the bond with an alkaline base forms, then the chemiluminescence is also shown. Then we have cleavage-based amplification. Cleavage-based amplification. Now this detects target nucleic acids by using a series of probes that bind to the target and then overlap. So it detects target nucleic by using a series of probes that bind to the target and overlap. So the probes overlap. Now cleavase is an enzyme that has been isolated from bacteria that recognizes overlapping sequences. So cleavase is able to identify overlapping sequences of DNA and makes a cut that cleaves in the overlapping piece. Now, in vivo, this activity is most likely important in repairing DNA. So, in vivo, this cleavage cutting thing happening in already formed strand is relevant when you have issues with repairing a DNA. The amount of signal can be quantitated and related directly to the amount of target molecules in the sample. This I expect you to understand. The signal can give an indication of the original target. So in this one, it detects a target nucleic acid by using a series of probes, and these probes have an overlap. Then cleavase is an enzyme that has the ability to recognize these overlaps and makes a cut in them. So that's hybrid capture assays and cleavage-based amplification. Now the cycling probe. A cycling probe, like the name says. In the cycling probe method of amplification, target sequences are detected using a synthetic probe, which consists of DNA, RNA, DNA. The probe's template is bound and released in a cyclic pattern, and each time a dye is released, which can be quantified. So the probe's template is bound and released in a cyclic pattern, so from one end DNA to RNA to DNA like that. And then each time in this cycle, it releases a dye. Now, this method has been used to detect genes associated with antimicrobial resistance in bacteria, such as methicillin resistance in Staph aureus, and then vancomycin resistance in Enterococcus. So, in cycling probe amplification, the target sequences are detected. Now, this detection is done by using a DNA, RNA, DNA combo. Now the probe's template is bound and released in a cyclic pattern. So we bind the probe's template and then it is released in a cyclic manner, and dyes are released for quantification. So sequencing, this is a technique of identifying the nucleotide arrangement in an RNA or DNA. So you want to see how they are arranged, the various nucleotides, gene, T, C, A, and all that in the RNA or DNA. So you want to see how they've been arranged. So if your name is Adomako, Adomako actually means Adom Ara Enko, only grace. So if your name is Adomako, then if Adomako is a gene, then you want to know how the letters have been arranged. So when you do a sequence, you know that Adomako is arranged as A, D, O, M, A, K, O. When you are successful in doing this, then sequencing has been done. All right. So DNA sequencing. So after amplifying a DNA target, so for instance, if you use PCR to amplify a DNA target strand, there is the need to identify the arrangement of its nucleotides. This is the relevance of DNA sequencing. So when you have done the PCR, yes, you have isolated a gene, but what is the arrangement there?
So DNA sequencing is the determination of the nucleotide arrangement in a portion of DNA. We also say that this sequence information, or the order in which the nucleotides are arranged, is used routinely for a variety of purposes, including detecting mutations. So even when there's a mutation, sometimes it can just be a single, or let me say 3 pairs of nucleotides that have been replaced or changed. So one of the purposes is to help you detect a mutation. Now type in microorganisms. So you have a series of microorganisms. You want to know whether A and B are the same. Sequencing can help. Then identifying human haplotypes. If it were to be a live person, I'll ask you what a haplotype is. So just find out what haplotypes are. And then designating polymorphism. Now find out what polymorphism is. That's why I've highlighted these ones. So check on what they are. So that's DNA sequencing. Now there are direct and indirect methods of identifying the DNA sequence, but the direct methods are the most useful or the most helpful. With a direct determination of the order or sequence of nucleotides in a DNA polymer, it gives the most specific and direct method for identifying genetic mutations or polymorphisms when you do the direct sequencing. So when looking for changes affecting only 1 or 2 nucleotides. So if you do the direct one, you can identify the changes that are found in just a single or 2 nucleotides that have changed, and just that one can cause a mutation. So that's a relevance of the direct sequencing method. So I said this one, there are direct and indirect methods of identifying DNA sequence, but the direct methods are useful. Now, the direct methods can be done manually or using a fully automated system. So you can do it manually or use a fully automated system. Now, there are 2 manual techniques that are used. 2 manual techniques that are used, or 2 DNA sequencing manual methods. We have the Maxam-Gilbert method, that's made up of 2 people, and Sanger method, the Maxam-Gilbert method, and then the Sanger method. You need to keep this in mind. Maxam-Gilbert and then Sanger. So the Maxam-Gilbert method was developed in 1970s by Alan M. Maxam and Walter Gilbert. Now it requires either a double or single-stranded DNA portion that is going to be sequenced, and then one of the ends is radioactively labeled. So the procedure for sequencing to be done, the labeled fragment, that is radioactively labeled fragment, or the template that is being used, is allocated into 4 tubes. So tube 1, tube 2, tube 3, tube 4, you put the fragment in there. Now each aliquot is treated with a different chemical, with or without high salt. Now when you add a strong reducing agent such as 10% piperidine, the single-stranded DNA will break at specific nucleotides. Now, after the reaction, the piperidine is evaporated, and the contents of each tube are dried and resuspended in formamide for gel loading. Now, the fragments are then separated by size on a denaturing polyacrylamide gel. So you are going to do electrophoresis over there. Now, the electrophoretic process helps to determine the sequence by exposing the dried gel to light-sensitive film and develop for viewing. Don't forget you have radioactively charged the ends, so you can view under light-sensitive film. Now, it is a relatively efficient way to determine short runs of sequence data, but then it can't work for long fragments. So don't forget Maxam-Gilbert is good for short data or short runs, but for long fragments, you are in trouble. You will not succeed in getting it. Now, in addition to, we also use hydrazine. The hazardous chemicals hydrazine and piperidine require more elaborate precautions for use and storage. So if you are using this method, then be ready that you are exposed to hydrazine and piperidine, and these can have an effect on you. So this method has been replaced by the di-deoxy chain termination sequencing method. And that is a better way of going about this procedure. So now we come to di-deoxy sequencing, or the Sanger method. The Sanger method. Now, the original di-deoxy chain termination sequencing methods required a single-stranded template. Templates up to a few thousand bases long could be produced using M13 bacteriophage. That's a bacterial virus with a single-stranded DNA genome. So we could produce a sequence of about few thousand bases long with this bacteriophage. That's in the original system. Now the di-deoxy chain termination sequencing is a modification of the DNA replication process, and you know DNA replication by now. So a short synthetic single-stranded DNA fragment, which we are calling a primer, which is complementary to sequences just in the 5' region of the DNA to be sequenced, is used for priming di-deoxy sequencing reaction. So you need a primer to initiate the whole reaction. Now, for detection of the products of the sequencing reaction, the primer may be attached covalently at the 5' end to a 32 or a phosphorus 32 labeled nucleotide. That's what I mentioned earlier. Or a fluorescent dye-labeled nucleotide. So you are either using a phosphorus labeled or a fluorescent dye-labeled nucleotide. Now, just like in DNA replication, the nucleotides are added to form a polymerization of the deoxynucleotides to make full-length copies of the DNA template polynucleotide. So you have a strand of the DNA, you replicate it to form a polymerized deoxynucleotide structure. Now this modified dideoxynucleotide, DD, dideoxynucleotide phosphates, DDNTP, are added to the reaction mixture to help terminate the polymerization chain. So if you don't terminate it, it will keep replicating or producing. So you need the DDNTP derivatives to cut short this polymerization reaction. And then your almighty thermocycler comes in. This is used to repeat this cycle because you know that in DNA replication, which is, let me say, exemplified by the PCR, you need a thermocycler to repeat the system. Heat, cool, heat, cool, heat, cool, heat, cool, like that. So after the reaction is done, the fragments are run through a long, thin tube containing a gel matrix in a process called capillary gel electrophoresis. So you are doing electrophoresis, but this one is a capillary with a gel, and it's a long tube. So you run your fragments through this one. Now, short fragments move quickly through the process of the gel, while long fragments move slowly. And this one, if you've done electrophoresis, you know that mass is key to the movement of particles, mass and charge. But in this case, mass is key. So the very light or the less heavier ones move through quickly, while the heavier ones or the longer fragments move slowly through it. Now, as each fragment crosses the finish line, at the end of the tube, it is illuminated by a laser. So the thing has been dyed, it is illuminated by a laser, allowing the attack dye to be detected. So they are all passing out through a finishing line. And then there's an illuminator or a laser which is causing illumination and helping identify that particular fragment that has come out through the output of this capillary gel electrophoresis chamber. Now the smallest fragment, that is ending in just one nucleotide after the primer, crosses the finishing line first, followed by the next smaller fragment ending 2 nucleotides after the primer, and so on and so forth. So the one that is just one nucleotide, if the nucleotide say 4 or 6, the primer, sorry, is 4 or 6 nucleotides, the next thing that will pass, or the first that will pass, will be one that has just a nucleotide more than the primer, so 7. Then the next one will be the one that has just 2 nucleotides more in that order. So thus, from the colors of dyes registered one after another on the detector, the sequence of the original piece of the DNA can be built up one nucleotide at a time. The data recorded by the detector consists of a series of peaks in fluorescence intensity and shown as what we call a chromatogram. The DNA sequence is read using these peaks. Let me just make it simple. So we want to sequence the gene Adomako. And I've told you the gene Adomako codes for Adom Aranko. Now, if my primer is 4 nucleotides, so ADOM is the first set of the primer or the nucleotides of the primer. When I go through this Sanger dideoxysequencing process, I'll produce a chain which is 5 letters. So Adom is ADOM4. The 5th letter in Adomako is ADOMA. So my next fragment will be ADOMA. Then the next fragment what I will come will also be ADOMAK. Then the next one will be ADOMAKO, Adomako. So when they are falling out, the ADOMA, which is just a nucleotide more than the primer, would come out. Then afterwards, ADOMAK would come out. Then finally, ADOMAKO would also pop out. So they will be in that order in terms of length of fragments. Now, we have spoken about manual methods. We are talking now about automated methods. Now, the automated fluorescence sequencing. This procedure has incorporated automated systems to do cycle sequencing, electrophoresis, and reading of the sequencing ladder. So, for example, in the past, if you wanted to read the HB, you need your DraftKings solutions. You manually pipette. You put it into your either colorimeter or spectrophotometer, whichever is available, and you search for that. You take the reading, then you calculate. Now, in a fully automated hematology analyzer, you just get your sample, you aspirate, and then that is done. So in the same manner, if you want to automate this whole sequencing thing, then it should be able to do the cycling by itself, the electrophoresis by itself, and then reading of the sequencing ladder, the one I just did with Adomako, it will read that one too by itself. So it's an automated fluorescence sequencing. Now we are saying that a requirement for this automated reading of the DNA sequencer ladder is the use of fluorescent dyes instead of the radioactive nucleotides. So in this case, we are using fluorescent dyes instead of the radioactive nucleotides to label the primers or sequencing fragments. Now, these fluorescent dyes used for sequencing have distinct colors or peak wavelengths of fluorescence emission that can be distinguished by automated sequencers for each of the 4 nucleotides. So every nucleotide is coming with its own color or peak that can be read. So now we come to hybridization and expression analysis. So when we talk about hybridization, so we are talking about the process which combines 2 single-stranded forms of DNA or RNA molecules and then allowing them to form a double-stranded molecule. So 2 single-stranded coming together to form 1 double-stranded. That's the whole concept of hybridization. So when you see DNA hybridization, don't think beyond what you already know in DNA. It is either it is combining 2 single strands to form 1 double strand. All right. So DNA hybridization involves the formation of a double-stranded nucleic acid that is either a DNA double helix or an RNA that is supposed to be DNA underlined red. So it's RNA DNA duplex. So you can either form a double-stranded DNA or a double strand of 1 RNA and then 1 DNA, forming an RNA DNA duplex. Now, this hybridization process is accomplished by denaturing the DNA that is to be analyzed, adding a labeled probe, and then incubating the reaction. So you break it up, bring a labeled probe, you join them. So DNA hybridization allows the identification and cloning of specific genes. So you are able to identify and then you clone specific genes. That is DNA hybridization helps achieve that. Also helps in the analysis of levels of mRNA in cells. So if you want to know the levels or the quantums of mRNA in cells, the procedure can also be useful. And then analysis of the copy number of sequences in the genome, and then DNA fingerprinting. So how many copies of a sequence do we have? We can analyze that. And then also with DNA fingerprinting. We'll come to fingerprinting later. So now let's look at nucleic acid hybridization. So like I said, hybridization or annealing is based on the ability of a single-stranded nucleic acid sequence, that is either a DNA portion or an RNA portion, to specifically bind to its complementary strand. So the hybridization or annealing is the ability to join one single strand to another single strand. That is easy put. Now, if a dsDNA, DF is double-stranded. When you see DS, it is double-stranded DNA. When you see S, a single strand. So if a double-stranded DNA is denatured or broken up or melted, it will hybridize when the denaturant or heat is removed. So once there is the heat, it's broken up. When the heat or the material denaturing is taken away, it joins back. Now, a few bases start the process, and the rest of a matching sequence will hybridize much like a zipper closing. So when the heat is taken away or the denaturant is taken away, it zips back. It zips back. So one will join like that, then it starts increase like you are closing your zip. So the next one is that hybridization in molecular methods uses single-stranded nucleic acid probes. So you use single-stranded nucleic acid probes. It can either be DNA form or RNA, or oligonucleotides of defined sequence. So you're either using single-stranded nucleic acids or oligonucleotides of defined sequence to hybridize to a target DNA or RNA of interest. So there's a target, you form a particular nucleic acid single strand, then you connect it to that portion. Now the probe can be labeled with enzymes, chemiluminescent, radioisotopes, magnetic particles, or fluorescent moieties that can be readily detected or captured by automated instruments. So you want to find out which name combines with adomako. Then you realize that, oh, quabia is a perfect combination to adomako. You introduce quabia into a system where adomako is found. Before you send the quabia, you label the quabia with either an enzyme, chemiluminescent, a radioisotope, magnetic particles, or a fluorescent material. When quabia finds adomako and binds, then your magnetic or whichever probe which has been labeled can now be detected by an automated system. Now, complementarity between the two strands is determined by the formation of specific hydrogen bonds between nucleotide pairs or bases. And you know that they work in pairs. Such that only the base pairs adenine thymine, adenine uracil, that is RNA, DNA, RNA, and guanine cytosine form hydrogen bonds. So you need complementarity, and it is only bound within these, giving sequence specificity to the double-stranded duplex. And as you know, if it is not having a similar thing, it will not bind to it. So this complementarity is key for a hybridization to take place. Now, the degree of binding and specificity of the binding depends on a number of factors. So the degree to which it's bound and then the specificity of the binding depends on a number of factors, which control the strength of the hydrogen bonds between the complementary nucleotide bases. And these include the temperature, the pH, the denaturant that was used as a formamide, salt concentration of the buffer solution. So these are key to determine how strong the bond is. Now, however, in DNA hybridization, we can accept a certain number of mismatch base pairs. So some portion or to an extent is tolerated. So for example, the base adenine lining up opposite to guanine would be a mismatch because G against C is not G against A. However, this can be to an extent allowed. As long as a significant number of base pairs do match and form bonds, the strands will still hybridize. So for instance, you are closing your zip. You know, sometimes your zip, if you don't get a good one, some portion cannot lock. When you're able to bypass that and go to the end, your zip is still closed, although somewhere in the middle, there isn't a close fit. So that's what we are talking about here. There can be a mismatch, but the level of mismatch should be reasonably small. Now, the degree of mismatching tolerated in hybridization reaction is called stringency. So the extent to which we allow mismatch in hybridization is called stringency. So when you see stringency in a test or in an exam, you should be able to tell me what stringency means. Then the next point. It says that hybridization methods are currently in use for the detection and identification of pathogens directly from clinical specimens. So now we can use hybridization methods to detect and identify pathogens from clinical specimens. So directly in the specimen, we can do hybridization to identify a specimen. Now, so in situ hybridization, just the word in situ means established in it or centered in it or found in it. So in situ hybridization. Now this brings a DNA or RNA probe into contact with the target DNA or mRNA in the tissue or cell under investigation. So you are not breaking the cell to release the DNA before you identify what the DNA contains, but you want to bind the DNA or RNA whilst it's in the tissue. You want to bind it to something. That is in situ hybridization. Now the technique finds use in the histotechnology study where tissue sections are probed for the presence of an infectious agent or often viral agent. So you want to find out if a particular cell has been infected by a particular virus. You can use in situ hybridization where you don't first break up the cell before you determine whether it is bound to something. You, whilst in the cell, you bind it to that determinant or the probe and see that yes, this virus is actually inside the cell. Now the modified form is the fluorescence in situ hybridization. That is FISH, F-I-S-H, fluorescence in situ hybridization, and useful in cytogenetics. Now the next one, the fixation and preparation of the tissue must preserve nucleic acid, and the probe must have access to nucleic acid during the hybridization period. Formalin fixed paraffin embedded tissues may be used for hybridization reactions. So formalin fixed paraffin embedded tissues. So the tissue has been fixed by formalin and then the paraffin has been embedded in it. May be used for hybridization reactions. The tissue is fixed to the slide and the paraffin removed by usual means. So after fixing and then embedding the paraffin, you remove the paraffin. A digestion process with hydrochloric acid or proteinase is necessary to remove histones and other proteins that may mask nucleic acid. Remember the histones, the DNA winding up around something. Remember that, histones. The section is subjected to heat, about 85 degrees Celsius for 10 minutes so that it denatures. A solution containing the probe is added and the preparation is allowed to sit overnight for hybridization to take place. Non-hybridized probe is removed by washing the sliding buffer. So like you do for your TB slides, you are trying to stain the acid-fast bacillus. You stain with something, you wash off the remaining. So it's using a similar technique or understanding to do this fixation and preparation of the tissue. So you can see 3 pictures here. You can see A, the chromosome showing fluorescence, B, cells showing fluorescence. So the chromosomes showing the fluorescence are there. Then the right one, you have the cells showing the fluorescence, meaning that they've been able to bind the labels or the probes into the cell. And that's what you are seeing on the right side. Now, enzyme labeled probes are common for this technique. Using the biotin avidin horseradish peroxidase label or alkaline phosphate label. The appropriate chromogenic substrate is added to the preparation and labeled probe is located in tissue under microscope examination. So you label it and you observe it. Fluorescent label probes are also extremely common and only a fluorescent microscope for detection. So fluorescent label probes are available and you need a fluorescent microscope for the detection. Digital images are taken in each color and then the images are combined. So you take these digital images in different colors and then combine them to form your fish fluorescence in situ hybridization. Now we come to nucleic acid blotting. Nucleic acid blotting. Now blotting is a method of putting DNA or RNA or protein onto a membrane for studies and detection. So you pull the DNA and put it onto a membrane, onto glass, onto anything. You are putting it on a membrane. Usually prior to blotting, the molecules are separated based on size or mass by electrophoresis. And that's true. Before you can undertake these blotting techniques, you need to identify the masses using electrophoresis. So nucleic acid blotting. So blotting is a method of putting DNA, RNA, or protein onto a membrane for further studies and detection. So if you want to do further studies, you put the DNA, RNA, or the protein onto a membrane, and that is blotting. Now, usually prior to blotting, the molecules are separated based on size or mass by electrophoresis. Then the different types of blotting techniques will come in. So there are 4 different variations of blotting that we'll look at: northern blotting, southern blotting, western blotting. And don't be tempted to say eastern blotting. We don't have eastern blotting. We have southwestern blotting. So the north is there, the south and the west is there, and then we have the south and west combined. So these 4 we are going to look at. So let's first talk about the southern blotting. Southern blotting. So Greater Accra blotting. Don't get right Greater Accra blotting anymore. It's southern blotting, please. Okay, now this is used to identify specific sequences on target DNA. So specific sequences on target DNA using labeled probes which are complementary to a DNA sequence in the sample. Now this is named after Edwin Southern, who was the first to report the procedure. And sometimes as lab personnel, we shouldn't ignore the little skills and techniques we pick on the bench which become a standard that we use and it's working. You can write a paper on that and then it will be named after you. So very soon you would hear Kwabia blotting or Adumako blotting because I've developed another technique for blotting. All right. Now in the southern blot, DNA is isolated and cut with restriction enzymes. So you first isolate from your cells, you cut them with restriction enzymes. You cut them with restriction enzymes. Now the fragments are separated by gel electrophoresis. dupyrinated and denatured, and then transferred to a solid support such as nitrocellulose. So you first isolate them from the cells, now cut them restriction enzymes, break them into fragments on a gel, denature them, and then transfer them to a solid support such as nitrocellulose. Now the final step of the procedure, the DNA fragments are exposed to a labeled probe, which we are calling a complementary DNA or RNA. So there's a labeled probe, DNA fragments. You expose them to an originally labeled probe, which we are also calling a complementary DNA or RNA. That is specific in sequence to the region of interest. The unbound probe is removed, and the signal of the probe is detected to indicate the presence or absence of the sequence in question. Now, the original method entailed hybridization of a radioactively labeled probe to detect the DNA region to be analyzed. As long as there's a probe of known identity, this procedure can analyze any gene or gene region in the genome at the molecular level. So what are we saying? You have a probe that has a specific sequence and has a particular region of interest. Now, when you remove this unbound, you bind some of the probe and then some will be unbound. When you remove the unbound one, you can identify a signal from the probe that is present in your target of interest or absence. So it helps you identify whether it is present or absent in your system in question. Now we come to the northern blotting. Northern blotting, let me just repeat myself on the southern blotting, or generally blotting. So in blotting, you cut a particular DNA into fragments, you pull them over an electrophoretic gel to expose them and know which of them is relevant. Then you pick your probe and go and look for it. So it's like you are looking for something inside the DNA. Generally, that's what the blotting thing comes. Now, what you are going to use to look for that thing is what we are calling a DNA probe. But it has to be labeled. If you don't label it, how would you know that, yes, this portion is where my thing went to attach itself to? So it's like a magnet. I'm going to look for a metal piece. That's the whole concept of the probe. Sorry, the blotting. So earlier we spoke about the southern blotting, which is specific to DNA, and we are now talking about the northern blotting, which is specific to the RNA molecule. The northern blotting, which is specific to the RNA molecule. So it is used to identify a specific RNA molecule in a mixture of different RNA by using a probe, a DNA probe. Now, the northern blot is a modification of the southern blot technique and was designed to investigate RNA structure and quantity. Although most northern analyses are performed to investigate levels of gene expression, that is the transcription from DNA, and stability, the method can also be used to investigate RNA structural abnormalities resulting from aberrations in synthesis or processing, such as alternate splicing. So apart from it helping you to identify the structure and quantity, it also helps you to find out whether transcription went on properly. Now, splicing abnormalities are responsible for a number of diseases such as beta thalassemias and growth hormone deficiency. So you see how relevant your northern blot is. Analysis of the RNA structure and quantity indirectly reveals mutations in the regulatory or splicing signals in DNA. So if there are issues with the signals that the DNA is bringing, this northern blotting, which is concerned with RNA structure and quantity, can give you an idea of the fact that there's something going on wrong over there. Then we come to Western blotting. Western blotting. Now, Western blotting is a modification of the Southern blotting. Western blotting is a modification of the Southern blotting, and it is used for identifying specific protein molecules. So we move from DNA to RNA, now we are talking about protein. So used for identifying specific protein molecules using protein probes. So a protein probe goes to look for a particular protein, and that's with Western blotting, that are labeled with a fluorescent dye or with a radioisotope such as sulfur 35. So Western blotting, talking about proteins. Now, the most widely used protein probes for Western blots are antibodies. Don't forget, an antibody is a protein. This can either be a monoclonal or polyclonal form of an antibody. These are used to detect target proteins, which are called antigens. So they are antigens, and you are bringing antibodies to go and look for them. So if that antibody is present and the antigen is present, the antibody which we have labeled will be able to bind to it. There are many variations on the Western blots. Generally, serum, cell lysate, or extract is separated on SDS polyacrylamide gels. Last time I asked about SDS pH. That is SDS polyacrylamide gels. Go find out about the SDS portion of it. Or isoelectric focusing gels, IEF. So you can be dealing with serum, cell lysates, extracts, which will be separated on these gels. Now the former resolves proteins according to molecular weight and the latter according to charge. So SDS polyacrylamide gel, that's with molecular weight, then isoelectric, that's separate with charges. So that's Western blotting. Then now Southwestern blotting. Southwestern blotting. A combination of the Southern blot and Western blot techniques is also used in the characterization of specific DNA binding proteins. And this technique is called Southwestern blot, since it involves hybridization of both protein and DNA. DNA we said is south, protein we spoke about as west, north is RNA. So southwest is proteins and DNA. Now short regions of the target DNA sequences are labeled and then serves as probes for hybridization reactions. So there's a target DNA sequence that is already labeled and becomes a probe. Now many DNA binding proteins can be characterized by this method. DNA binding proteins can be characterized by this method. So basically this one is able to deal with DNA and protein mergers or bound molecules. So this is with Southwestern blotting. I think I'll need to get you videos on these that we've mentioned. I said there's no eastern, there's south, there's north, there's west, and then southwest. So I'll get you videos, short videos on these so that you can understand the procedures that are involved. So we move on. Now we come to gene expression analysis, gene expression. So you want to know what the output of a particular gene is. How do you go about it? Now, array technology can be applied to the gene analysis or DNA analysis by performing comparative genome hybridization and then leading to gene expression. And these are done on expression arrays. So in the array technology, you are looking at how you can cause a particular gene to be expressed. Then that is in a form of a protein, and then you find out what it is doing. There are several types of these array technologies. We have microarrays, macroarrays, high-density oligonucleotide arrays, and microelectronic arrays. But we will not look at all these. We will just look at the microarrays. So the DNA microarray assays. A DNA microarray is a collection of microscopic DNA spots attached to a solid surface. It's one of the newest ways to analyze a lot of samples with one probe or a lot of probes spotted with one sample. So a lot of samples with one probe or a lot of probes with one sample. So you send, say, 5 probes into one sample or 1 probe into 1 probe into 5 samples and 5 probes into 1 sample, so the reverse. Now, these probes usually correspond to a short section of a gene, which is generally at the 3' end. Each microarray includes 1 or a few probe sets for each interrogated gene. Interrogated gene because we know what that gene does. These are used to hybridize to a cDNA sample, which is the target, under high stringency conditions. The probe target hybridization is usually detected and quantified by detection of the fluorophore, that's silver or chemiluminescence labeled targets to determine the relative abundance of transcripts in that target sample. So let me make it simple. You know a gene, you know what it does or what it expresses, then you go looking for it in particular genome or a pool of sample. Now, DNA arrays are hybridized exactly as dot plots if the array is on a membrane, and as an in situ if the array is on glass. So if it's on glass, you do in situ, and then if it's on a membrane, you do the dot plots. Now you need a special software to analyze the huge number of data points that we have available. So you have data set in colors, and you can't use your eyes to do it like you do with your regular, let me say, malaria microscopy. No. Now we have 2 color arrays which are used to determine changes in the expression of genes. Now cDNA, complementary DNA, is made from normal and test cells, and then we hybridize them to genomic arrays. Now the relative increase and decrease in the expressed genes can be determined. So whether the genes have increased or decreased, you can determine because of the color arrays that are developed. This technology may remain the domain of research for some time, but clinical applications include rapid analysis of expression of genes in tumors, in inflammation, and in many others. I take a sample, I have a gene that codes for, say, cancer. I can do my DNA microarrays on it and know that no, this cell has some cancerous genetic code in it, and I can start dealing with it. Okay, so now we come to molecular cloning. Cannot do your nucleic acid techniques without talking about molecular cloning. So we deal with molecular cloning. Now, molecular cloning. For example, you want to produce a vaccine, and then the vaccine is a particular protein. Now, if you identify that protein and you want to produce more of that, molecular cloning is relevant. If it's a DNA portion that you want to produce, molecular cloning is relevant. So cloning is just like producing copies of oneself. That is cloning. So DNA cloning is the process of making multiple identical copies of a particular piece of DNA. Now, in a typical DNA cloning procedure, the gene or other DNA fragment of interest, which is perhaps a gene for a medically important human protein, is first inserted into a circular piece of DNA called a plasmid. Now, the plasmid is a circular DNA found in some bacteria. The insertion is done using enzymes that cut and paste the DNA, and it produces a molecule which we are calling a recombinant DNA, a recombined DNA, or DNA assembled out of fragments from multiple sources. So you bring different DNAs from different sources and you merge them. So let's look at the steps that are relevant in DNA cloning. So DNA cloning is used for many purposes, and we've mentioned one above. As an example, we see how DNA cloning can be used to synthesize human insulin in bacteria. So these are the steps. The registration enzyme cuts, the bacterial transformation, and then the protein production. So we come to DNA cloning. Now the steps in this. Step 1, restriction enzyme is used to cut. So a restriction enzyme is a DNA cutting enzyme that recognizes a specific target sequence and cuts DNA into 2 pieces at or near that site. So it identifies a specific target and then it cuts. Many of these restriction enzymes produce cut ends with short single-stranded overhang. So it cuts at a particular point and there's an overhang. If 2 molecules have matching overhangs, they can base pair and stick together. So if you have 2 overhangs which are similar, they can stick together. However, they won't combine to form an unbroken DNA molecule until they are joined by a DNA ligase. So a restriction enzyme cuts, a DNA ligase has to come in and then joins it and then seals the gaps in the DNA backbone. So the first step, we cut, and then when we see the relevant thing, we join by DNA ligase. Then the second step is bacterial transformation and selection. I mentioned plasmids earlier. So plasmids and other DNA can be introduced into bacteria, that is, such as introducing it into E. coli, which we have in our labs daily, in a process called transformation. Now, during transformation, specifically prepared bacterial cells are given a shock, that is high temperature shock, which encourages them to take up the foreign DNA. So when the shock comes, they open up and then the foreign DNA enters their plasmid. So after entering their plasmid, a plasmid typically contains an antibiotic resistance gene, which allows bacteria to survive in the presence of a specific antibiotic. So that's what the plasmid does. It helps it sustain itself in an antibiotic. So it has an antibiotic resistance gene. Thus, bacteria that took up the plasmid can be selected on nutrient plates containing the antibiotic. Bacteria without the plasmid will die, while bacteria carrying the plasmid can live and reproduce. So the surviving bacterium will give rise to a small dot-like group or colony of identical bacteria that all carry the same plasmid. So those who are carrying the plasmid, they survive, and then we can see a colony of these which can be identified. And the last point here, not all colonies will necessarily contain the right plasmid. That's because during a ligation, DNA fragments don't always get pasted, so some will not be pasted in exactly the way we intend. Instead, we must collect DNA from several colonies and see whether each one contains the right plasmid. And methods like restriction enzyme digestion and PCR can be used to check these plasmids, whether they are right. So you heat shock them in bacterial transformation. When they enter the DNA, you use the antibiotic selection factor, natural selection, and this artificial selection, to choose those which survive, and those colonies are dealt with using a ligation, a restriction enzyme, or PCR to check whether they are those that you need. So this is the second step. Then the final step is the protein production. So once we have found a bacterial colony with the right plasmid, we can grow a large culture of plasmid-bearing bacteria. Then we give the bacteria a chemical signal that instructs them to make the target protein. So this is what we have done. You pick a plasmid relevant with your relevant target gene, you insert it into a bacteria, then you tell the bacteria to produce proteins from that section of the gene which is relevant to you. So the bacteria serves as a miniature factory, churning out large amounts of protein. So for instance, if our plasmid contains the human insulin gene, the bacteria will start transcribing the gene and translating the mRNA to produce many molecules of human insulin protein. So at this point, think of your DNA, RNA, protein step. Think of this step. So your DNA is able to transcribe an RNA, is able to translate into a protein. So this is the step or the technique that is being used here. So you put in your gene for insulin, enters the plasmid, push it into the bacteria. Then you tell the bacteria to produce the protein corresponding to that particular gene. When that protein is produced, you now harvest it as your insulin. So the last point, once the protein has been produced, the bacterial cells can be split open to release it. And there are many other proteins and macromolecules that will be floating around in the bacteria besides the target protein. Because of this, the target protein must be purified or separated from the other contents of the cells by biochemical techniques. The purified protein can be used for experiments or in the case of insulin administered to patients. So that is molecular cloning or DNA cloning. So how or when do we use this DNA cloning? So in biopharmaceuticals, we mentioned insulin. So it's one of the ways of doing it. We can also use it for the human growth hormone, producing it. Still under biopharmaceuticals, tissue plasminogen activators, which are useful for strokes and which prevent blood clots. We can use it for gene therapy. So in some genetic disorders, patients lack the functional form of a particular gene. Gene therapy attempts to provide or produce a normal copy of the gene to the cells of a patient's body. So for example, DNA cloning was used to build plasmids containing a normal version of the gene that's non-functional in cystic fibrosis. When the plasmids were delivered to the lungs of the cystic fibrosis patients, lung function deteriorated less than quickly. In cystic fibrosis, fluids are filling the lungs, but once you introduce the normal gene into the lungs now deteriorate less, and then the cystic fibrosis becomes lesser. So that's a direct gene therapy. Then we can also use molecular cloning for gene analysis. So in basic research labs, biologists can often use DNA cloning to build artificial recombinant versions of genes. That helps them understand how normal genes in an organism function. So you cut the gene, put it into a recombinant version, into a bacteria, reproduce it, and you are able to study its usefulness. Now, gene library. So the word gene, you know, the word library, you know. So a gene library is a library of genes, simply put. So what's a gene library or genomic library? So a genomic library is a collection of overlapping segments of genetic or genomic DNA which are cloned into a backbone vector, which statistically includes all regions of the genome of an organism. If we take the Human Genome Project, we are trying to get all the genomic arrangements of the human being. So the resulting cloned DNA is then transferred into a suitable host cell line. So we are trying to build the genome of an organism. So construction of a genomic library is an important initial step in many genetic studies or in the isolation and cloning of genes from an organism. Screening of genomic libraries can be useful in identifying genes of interest to medical field and then the biotechnology industry, as well as finding genes related to particular cellular functions. So when we build a gene library, we're able to know what each gene does, and then we will be able to deal with it with respect to research. So additionally, creating a representative genomic library of an organism is a prerequisite for genomic mapping or a complete genome sequencing. Genome sequencing is knowing the individual nucleotides in a genome. So this is the end of these two lectures, 5 and 6. I'm hopeful that the video will be useful. It's 2 minutes and about 2 hours, sorry, 2 hours and about 6 minutes. I pray that you break it into small sections, watch through, and then try and understand it. I'll send the links for the videos later. Thank you very much for your time. See you in the next lesson.